AP Biology 2.11, Origin of Self Compartmentalization. In this unit, this section, you will practice argumentation, that you have to support a claim with evidence for biological principles, concept, processes. The learning objective is describe similarities and differences in compartmentalization between prokaryotic and eukaryotic organisms. The essential knowledge, we distinguish different cell types based on whether they have nucleus or not. We distinguish prokaryotic cells that do not contain nucleus, and we have the eukaryotic cells that they have nucleus. That is the nucleus in here, okay? This big one. Cells that have nucleus or had a nucleus in their lifespan, I'm thinking about the human red blood cells now, that in our circulation, it does not have a nucleus anymore because it lost it in its development in the red bone marrow, but we still consider the red blood cell in our body as a eukaryotic cell. In addition to the lack of nucleus in the prokaryotic organisms, they are lack of membrane-bound organelles. Membrane-bound organelles in here is the mitochondrion, the endoplasmic reticulum, rough and smooth, and if I would draw, the Golgi apparatus would be there, the lysosome would be here. So the prokaryotic cells don't have anything like that. But let's not think about that prokaryotic cells are disorganized cells. Prokaryotic cells contain, for example, a nucleoid region where they have their circular DNA, compared to the prokaryotes who have linear DNA in their nucleus. I would like to point out both of the cells contain ribosomes. The ribosomes in prokaryotes, though, are a little bit different from the ribosomes in eukaryotes, but they are important sites for translation in the process of protein synthesis. Eukaryotic cells maintain internal membranes in the form of organelles that partition the cell into specialized regions. They have a membrane-enclosed nucleus where the DNA is, as I said, linear instead of circular in its structure. Both of the cells also contain cell membrane. The cell membrane and all of the membranes are made up of phospholipid bilayer, and they separate the outside from the inside. Now, evolution of the endomembrane system, here is this going to be endomembrane system, is really interesting. Please notice how the nuclear membrane, here is the nucleus, now the nuclear envelope is going to form like the infolding invagination of the plasma membrane around the nucleoid region, and later on it forms a nuclear envelope. But it happens like the invagination of the cell membrane also produces the endoplasmic reticulum and the Golgi, as well as the lysosome. It comes from the outer membrane of the cell. The next objective is describe the relationship between the functions of the endosymbiotic organelles and their free-living ancestral counterpart. What does it mean? That the cell membrane-bound organelles, such as the mitochondrion, here is that, was a free-living organism way back, a couple of million years ago, a couple of hundreds of million years ago, as well as the chloroplast evolved from a previously free-living prokaryotic cells via endosymbiosis. Here is the mitochondrion and this is the chloroplast. They lived freely. They were prokaryotic organisms. The theory of the endosymbiosis, that they now live together, these prokaryotic organisms, in the eukaryotic organism cell, is supported by the fact as the mitochondrion and the chloroplast, they contain circular DNA, just as the prokaryotic organisms. They have their own ribosomes, and the ribosomes also resemble to the prokaryotic ribosomes. They also contain double membrane system, and that suggests that once they were free-living prokaryotic organisms and they were engulfed by a eukaryotic cell. However, the eukaryotic cell did not digest the foreign cell, prokaryotic cell, but formed a symbiotic relationship with them. And this is the end of this topic, the origins of cell compartmentalization. I hope you learned something today, something interesting and new. Have a good day.